Good evening. How is everybody tonight? You've already had your cookies and your coffee and your water. As a reminder, for those of you that are in this room, there won't be a break. So for those of you that are watching from home, you're going to have to actually hit pause or just get up and run really fast as I'm moving from one slide to the next. Those of you in this room, if you need to excuse yourself for a bio break, please feel free to do so. Just do so and then come back quietly. We have so much ground to cover this week, which is why there is no break, so I apologize. Also, there's a fair chance that if you have questions, I may not be able to get to those questions. This is a night where those of you that are watching online and have two moderators may get more questions answered than in this room. But we're going to set up a Post-it pad, a big Post-it pad outside. Write questions down if I don't get to them, and then I'll respond. I'll send out a frequently asked questions based on the content that comes back, and we'll send that out by email. So if you don't get a chance to ask the question tonight and here, and fair warning, you probably won't because we have so much to cover, I will get to those questions and respond back out. Before we get started, there are a few words of appreciation that I need to express. I told you on the first day that I am not a farmer or a plant breeder or a horticulturalist. So there are some groups that over a year ago I reached out to and started asking questions about crops and about chickens and about beef. And so I want to thank Bragg Farms. So Barbara and Alan are actually here with us for Biotech, so thank you all so much. I had a great time riding in the combine as well. Aviagen, Dr. Derek Emerson, and Randy Moody from Little Mountain Farm were really helpful in giving me background about poultry and about beef genetics. And much of what we've talked about is because of some of the content that they pointed me to. Let me also say thanks to all the Biotech 201 volunteers who week in and week out check you in, get you name tags, get you in the right place, put handouts in your hand. They do a fantastic job, and I'm really grateful that they make this run so smoothly. And then I have an education outreach team that is continually putting content in my hands and helping by taking work off me so that I can get everything prepped and ready to do nights like this. So their names are up here, and especially the ones that are in bold. So Daisy Price, Michelle Morris, and Madeline Lofton have been helping with the online chat and the moderation. And then Stacy Brewer, who you all know, who keeps me in the right place at the right time and gets you in here and checked in. And really, Biotech 201 and 101 run because of Mr. Brewer, and I just want to express my thanks to him as well. And then last but certainly not least, we are really happy to partner with the Jackson Center to be able to provide this space and a great place to meet and AV and especially cookies and water. So it's always a pleasure to get to work with them. So thanks to all these people on the screen. By the time you leave tonight, in your email inbox will be your certificate of completion and a little bit of information about a new project that we're crafting called the Alumni Association, which is open only to those folks that have gone through Biotech 101 and or 201. So watch your emails for some information and then your mailboxes for some additional bits of information as well. All right, here we go. Everybody buckle your seatbelts. We're going to talk about genetically modified foods. We're going to talk about what they are and how they're created. We're going to spend most of our evening talking about the questions that people ask about genetically modified foods and the concerns that people raise. And we're going to go through many of those questions, and I'm going to talk to you about both sides of the responses back to those questions. And then we're going to talk very briefly about some new technologies that are on the horizon. And I think that these, let's do that. Is that better? Yeah, there we go. I think that these new technologies are really valuable because they potentially change the narrative for how we think about genetically modified foods, but we'll see. If you've been here any of the last 4 weeks, we've talked a lot about genetic modification, from conversations about domestication and what we've done for the last 10,000 years, to how we've specifically selected certain plants and animals because they had characteristics and qualities that we were really interested with or intrigued with, to marker-assisted selection and how we're now using genetic information to specifically select crops or animal traits. And then we talked about this whole concept of induced mutation, where we dip seeds in chemicals that cause mutations in DNA, or we take seeds or plant material and we irradiate them to try to increase the likelihood of finding mutations or creating mutations that we think might be useful. So we've been talking about genetic modification for the last 3 weeks. But today we're going to talk about a specific form of genetic modification that really more appropriately is genetic engineering. And genetic engineering is incorporating the genetic information from one organism into the DNA of a completely different organism. And this was until, without this technology, you would be talking about two organisms that normally don't interact. They don't breed, they don't share information. So there would be no way that without these technologies you would get information from a bacteria into a corn plant. So this is the World Health Organization's definition. Genetically modified foods are foods derived from organisms whose genetic material has been modified in a way that does not occur naturally through the introduction of a gene from a different organism. So that's what we're going to spend our time focusing on. This is a subset of the entire genetic modification conversation, but I want to frame it, even though I'm spending an entire night on it. We've been talking about genetic modification for the last 3 weeks. So that whole concept of what is natural and how do we make selections needs to be placed not simply against tonight, but about all these other things that we've covered. All right, so at this point, those of you here in the room and those of you watching at home, pull out or pull up the single-page insert. So in addition to the multi-page handout, you should have a single page that looks like this that talks about how are genetically modified crops developed. I wanted to make this large enough so that you could see all the content, so you weren't looking at something small. We're going to walk through this step by step, and the words that are on your handout are the words that are going to come out of my mouth, but they're not necessarily the words that are going to be on the screen because it's the way some of the animations work. So everybody got that in front of you? All right. So let's give the following example. Let's say that I want to create a plant that thrives in cold weather. And I'm going to start with my host plant. So this is the plant that I've got. It may have all these great qualities and characteristics, but the one thing that it doesn't do is it is susceptible to cold weather. It dies, it's not frost hardy, whatever the storyline is. But I want to figure out how to make my plant more cold tolerant. So one thing I might do that we've talked about before, I might look back throughout all the different varieties of this plant and look and see if I can find a similar version that is cold hardy, and then go through that traditional crossbreeding and those back crosses to bring that into this specific plant. Or I may create or look for mutations in this plant. I may look at a whole bunch of different versions of this and look for changes, or I may irradiate. I may go with the induced mutation approach and try to create cold hardiness. Those are all ways that I could try to incorporate cold hardiness. We're going to take a different route, and we're going to bring the gene that provides cold hardiness in from a donor. So I've got a jellyfish, and let's just say that this jellyfish grows and thrives in the Arctic, in the North Atlantic, in the cold North Atlantic Sea. And one of the reasons it does that is because it has a specific gene that scientists have identified, and this is all purely hypothetical at this point, a gene that scientists have identified that allows its cells to grow well under sub-zero conditions. So the cells don't freeze. The water in the cells doesn't turn into ice. So that's the storyline now. How do I get this information out of my donor? Here are my jellyfish cells, and here's the donor DNA. And we've done enough genetics on this. The sequence of the genome has been obtained, and we've done all the research. So we know that right here is the gene that confers cold hardiness in my jellyfish. So the first thing I need to do is actually isolate or clip that out of the rest of the DNA. So there are molecular tools, there are small enzymes that can cut the DNA in very specific places. And so I'm going to cut it out of my jellyfish DNA. And now that's the piece of DNA that I'm going to work with. And I'm going to add some switches, some start and stop genetic switches. So this is more genetic information. These start and stop switches actually provide the signals that say, okay, this gene is now going to be active in the specific cells I need of the plant. Maybe it's only going to be active in the leaves. Maybe it's going to be active in every cell of the plant. It's going to be active under certain conditions. When the temperature drops below a certain level, this gene is now going to be activated to produce the protein that gives it resistance, cold hardiness. So these switches, these are bits of genetic information, and by and large, they come from viruses and bacteria. We've studied these for years, for decades. We know many of the on-off or high-low switches in bacteria and viruses. And so we're going to add them, we're going to attach them to the cold hardiness gene. So now I've got all the important control information, and I've got something called a selectable marker, and I'll come back to what a selectable marker means. And I've got now to figure out how to get it into my plant cells, and I'm going to go through a two-step process. So there's a piece of DNA, it's a circular piece of DNA that's called a plasmid. And lots of bacteria have plasmids. It's one of the ways that bacteria share genetic information back and forth. This is not the main chromosomal information of the bacteria. It's additional circular information. And I can use the same set of enzymes that I cut the DNA out of my jellyfish, I cut the gene out of my jellyfish DNA. I can use a similar set of enzymes, and I can cut my plasmid. So it's no longer circular, it's open, and I can actually glue in my sequence. So now my on-off switches and my hardiness gene are in this circular plasmid. And then I can actually put it into a bacteria. Now, if you know students that take AP biology, this step of getting a plasmid into a bacteria is a routine process for them. It's called DNA transformation, and students in our high schools do DNA transformation all the time. It's a pretty straightforward process, getting a piece of genetic information into a bacteria. So we get our bacterial plasmid and we put it into our agrobacteria. Now, agrobacteria are one of the bacteria of choice for the following reasons. If you've ever had a plant that had crown gall, which looks like a whole mass of cells around the outside of somewhere on the stem, crown gall is caused by agrobacteria. So agrobacteria are really good at injecting some of their genetic information into the plant cells, and they cause the plant cells to grow rapidly in this kind of growth around the outside of the stem, and the plant cells produce nutrients that feed the bacteria. So agrobacteria is a great way to get genetic information into plant cells. So that's why we use agrobacteria. So agrobacteria inject their DNA into our plant cells, and we can look at that in a different version. Here's our host DNA. This is my plant's natural DNA. Here's the insert with my on-off and marker switches on it. What happens is it actually integrates in to the host DNA. So now I've got my cold hardiness gene and my on-off switches in the middle of my plant DNA. Now the problem is it inserts in random places. So if I did this 100 times, it would likely insert in 100 different places, or maybe 95 different places. So one of the things that has to be done that's part of the regulatory process is make sure that it doesn't drop in the middle of a really important gene. Because if it drops in and inactivates an important gene, that's problematic. If it drops in the middle of something that controls when a gene gets turned on and off, that's problematic. So I've got to make sure that I pick the right, I'll do this multiple times, and then I'll go back and say, where did it insert? Where did it insert? And I need to pick the one that inserts in a completely benign, quiet place. All right, so here's my cell that has now the insert with my cold hardiness gene and my on-off switches. And now I'm going to do something that's called tissue culture or tissue propagation. I'm going to take these cells and I'm going to grow them in a petri dish that has the right media, the right food that helps plant cells grow. Here's where that selectable marker piece that I talked about comes in. One of these on-off switches provides the cell with the ability to grow in the presence of a chemical that normally would kill the cell. Sometimes it's an antibiotic, sometimes it's something different. But it's a way for me to ensure that the only cells I'm going to work with are the cells that have taken up this insert. Because if they haven't taken up the insert, the chemical's going to kill them. If they've taken up the insert, one of the genetic switches lets them grow even if the chemical is present. So it's a selection to make sure I'm only getting cells with the insert. I grow that, I add hormones, I don't know if you can see this, but there's a teeny tiny plantlet right there, little roots, little stem and leaves. I then grow that and I get my entire plant. And because it has my cold hardiness gene in an ideal situation, in an ideal setting, and several $million in years later, through all this process and lots of regulation, I now have a plant that grows really well in cold temperatures. That is the process of creating a genetically modified plant. And if that makes your head hurt and you want to go to a happy place, that's okay. We're not going to cycle back to this a lot, but I want you to understand the science behind it. So all of the steps that we went through to get that genetic information out of our donor and into our plant cell, and then we have to make sure that it works, that it does what it says it's going to do. It's a pretty involved process, but all of these technologies have been used for decades. So if you know anyone that has diabetes, that has to take insulin, that insulin comes from a genetically modified bacteria. We took the bacterial DNA and we inserted the gene for insulin, for human insulin. And so when the bacteria reads the human gene, it reads it just like it would read any of its own genes, and it uses that information to make insulin, and the bacteria produce insulin, and we extract and purify it, and then that's what we do. So we've been, ever since we first created recombinant insulin, so we weren't getting it from pigs or cadavers, we've been using genetically modified organisms. I'm just showing you the same technology as it applies to a crop or to a plant that we would be looking at. The same technology could be used in an animal, so we're awaiting regulatory approval on a salmon that reaches maturity faster than the other salmon because it has a gene from another variety of salmon that lets it grow year-round instead of only growing in the warmer months. Now, it does not grow and grow and grow and keep on growing. Once it reaches maturity, it stops growing, but it does that in half the time. Same kind of concept. All right. Let me give you some examples of genetically modified crops, now that we've walked through the technology behind it. There are several genetically modified crops that are currently approved for growth. A handful of them are the ones that are most commonly grown. So this table talks about what plant we're dealing with, the gene that we've inserted from our donor, what donor we used, and what its purpose was. And you can see right here, the tomato, the very first genetically modified crop was something called the Flavor Saver tomato that came out in the 90s and gave tomatoes a longer shelf life, but these tomatoes actually tasted good. Well, relatively good. They tasted not as good as ones you grow in your garden, but better than, the hard as rock ones that you sometimes get in the middle of the wintertime. But the problem, there were a number of issues, not safety issues, not health issues, but a number of issues in terms of trying to grow enough and some of the financial business model. And so the Flavor Saver tomato was no longer available, but it really was the first genetically modified crop. So if you look at the rest of these, you're going to see some commonalities as you look down the purpose column. You're going to see insect resistance, and you're going to see herbicide tolerance. Those are the two most common traits right now in genetically modified crops. And one of the big knocks, one of the big arguments against the current set of genetically modified crops is that they really provide traits that are helpful to farmers, but they're not necessarily things that consumers would want or that consumers would notice. They're providing insect resistance and they're providing resistance to herbicides. We'll dig through what that means, but that is a fair statement. The criticism behind it may not be, but most of the existing genetically modified crops either are insect resistant or are herbicide resistant. And let me talk to you about what those two things mean. So the corn and the cotton and some of the other crops that I didn't show on here have taken a gene from a bacteria, Bacillus thuringiensis. Anyway, this bacteria right here. And this gene, or this family of genes, produces proteins that are toxic to certain insects, especially in their caterpillar stage. But they're not toxic to all insects. They're not toxic to many beneficial insects. But things like the cotton boll weevil or the corn borer the protein that this gene makes attaches to cells in their digestive system and actually prevents the digestive system from working, and the caterpillar die. Now, the protein, Bt protein, is actually a protein that's approved for organic use. So you actually, organic farmers put Bt, they either can spray it or they can put it in granular form around their crops, and the caterpillars eat it, and then it kills the caterpillars. The challenge is a lot of the caterpillars are on the underside of the leaves, or if they come to the leaf after it's rained and after it's washed off, then there are problems. So what they've done is they've taken this gene that makes this protein out of the bacteria and they've put it into the corn or the cotton. So all of the cells in the corn or the cotton plant or the eggplant or other plants produce this Bt toxin. Now, for you and I, and for most insects and for farm animals, we don't have the ability for that protein to bind to the cells in our intestinal system. It requires having a certain receptor, which is kind of like a baseball glove that sits on the surface of a cell that specifically recognizes the Bt protein, the toxic protein. We don't have that. So it does not affect our bodies, nor does it affect cows. Only this specific class of insects. So every cell produces this Bt protein, and when these caterpillars eat the leaves or eat the corn kernels, they ingest the protein, it attaches to the walls of their digestive system, and they die. So it is a built-in herbicide. Now, before some of you go, ugh, plants that make their own herbicide, yeah, you need to know that almost every plant makes its own herbicide. The natural defense mechanism of plants against insects is to produce proteins that are toxic to some of their pests, some of the things that try to eat them. So even if we're not talking about genetically modified corn or genetically modified whatever, if you eat a non-GMO ear of corn, you are still eating pesticides. It's just a different type of pesticide that the corn naturally produces. So just be aware of that. We would not be eating that pesticide if it was problematic for us. Well, we would not be eating it for long. So that's when I talk about Bt corn and Bt cotton, that's what I'm talking about. It produces this insecticide from this gene from the Bacillus bacteria. Now let's talk about herbicide tolerance or herbicide resistance. If you've ever sprayed glyphosate, which is the active ingredient in Roundup, on anything that is green, almost anything that is green, it kills it. It actually blocks a specific gene from carrying out its function of being able to make essential nutrients that the cell needs to survive. So the Roundup or the glyphosate, the active ingredient, blocks this action of making this essential nutrients. So that gene is called EPSPS. And corn has an EPSPS gene, and cotton has an EPSPS gene. But in those organisms, they are susceptible to Roundup. Roundup stops the function of the protein produced by the EPSPS gene. However, in a bacteria called agrobacteria, which is the same agrobacteria that we talked about as the vehicle that gets the gene into our plant cells on the previous slide, agrobacteria have a version of this gene, the EPSPS gene, that has a couple of genetic changes. It's got different alleles, different flavors. Maybe it's a C instead of an A or a G instead of a T in this gene, and it means that the protein this gene makes is resistant to glyphosate. So you can spray glyphosate, and this protein from this modified gene carries out its function totally normally, which means I can take this EPSPS gene from my agrobacteria, put it into a plant, and now the plant, in addition to making its own herbicide-sensitive EPSPS protein, is making the agrobacterium-tolerant EPSPS protein. There's a lot of P's and S's in this conversation. But it means that the plant doesn't die if I spray glyphosate on it, because the protein can carry out its function, this modified protein from the bacteria, in the presence of Roundup. So if I'm a farmer I can plant Roundup Ready, and different companies call it different things. Monsanto's version is Roundup Ready, but there are other versions, Herbicide Tolerant. I can plant this corn, or I can plant this, this soybean, or this cotton, and then when the weeds come up along with my plant, I can spray the field with Roundup or with glyphosate. And the weeds will die because they're sensitive, but my corn that now contains the bacterial version grows just fine. It tolerates it without any problem. So those are two key traits, Roundup Ready or herbicide tolerant, and BT or insecticide. So lots of what I'm going to talk about come back to those two examples. But there are other versions of genetically modified crops that bring in other genes, like the papaya, right here, that actually uses a bit of information from the papaya ring spot virus that makes it resistant to that virus. Or there's a version of corn that actually uses a protein from a bacteria that actually controls water release, that helps make the corn drought tolerant. This is a completely different version from the artesian corn that I told you about 2 weeks ago. That was chosen by selectively breeding specific versions of natural corn genes. This is made by genetic modification. But there are lots of other varieties that are in the pipeline as well. But most of our conversation is going to center around either BT or herbicide tolerant versions of genetically modified plants. And you actually can stack those. So I actually can create a plant that's both BT, that's producing BT, so it kills the corn borer or the cotton boll weevil, and it's resistant to glyphosate. And I can create one that's actually resistant not only to glyphosate, but it's resistant to glufosinate, which is a different kind of herbicide. So I can stack traits. So I can have a genetically modified plant that has multiple kinds of traits. All right. In 2014, 181 million hectares, and a hectare is about 2.5 acres. So lots and lots of land were planted around the world with genetically modified crops. They were planted in 28 countries. The ones in orange are the developed nations, 8 developed nations that plant genetically modified crops, and there are 20 developing nations, and those are shown in green. And you can see from the icons the different kinds of plants. So the corn or the soybean or cotton or canola. And it gives you a sense of where these are planted. Now you might notice that there are some European countries here that actually are approved for planting maize, herbicide-tolerant maize. These are for animal feed under pretty strict controls, but there are some European nations that have approved that. By and large, most of Europe has stayed away from genetically modified foods. There are some indications that some regulatory bodies are willing to look at that differently, but that doesn't necessarily sit well with many members of those nations, which feeds into a conversation we'll have in just a second about the controversy, or at least what both sides say about the controversy. And you can see the 4 most common crops are on this pie chart behind me. Soybean, which accounts for almost half of all genetically modified crops planted, and that is primarily herbicide-tolerant soybean, and then maize, corn, and then cotton and canola. So those are the 4 that make up the vast majority of the genetically modified crops that are planted around the world. And there were some new ones that were introduced last year. So we'll talk a little bit at the end about a specific potato that resists browning when you cut it. There's a drought-tolerant sugarcane that's been approved in Indonesia, and an eggplant that is BT eggplant, so it is resistant to the weevil, that has been approved in Bangladesh. Here are the 28 countries and specific listings of the millions of acres of crops that are planted in those specific countries. The most genetically modified crops are planted in the United States, which plants almost twice as much as its nearest competitor, not competitor, its nearest country, which is Brazil. And then you can see that in the United States, 93 to 96% of the corn, soybean, and cotton crop are genetically modified. So if we're looking at a field of corn, especially a field of corn for feed, and increasingly a field of corn for biofuel, and we're beginning to see fields of corn for sweet corn for eating, odds are it's genetically modified. If you are at the grocery store and you buy processed food, so you buy corn chips, or you buy tortillas, or you buy something that has corn syrup in it, odds are there's a really good chance that it came from a corn plant that contains the Bt gene that makes it resistant to the corn borer worm, and it probably was herbicide tolerant as well. Now, if we went to Europe and went shopping in the grocery store, we would see a very, very different set of things, because in Europe, by and large, genetically modified foods are banned from the from the food chain. So a clear difference right there, right off the bat.
The gray color in the previous slide, was that for sugar beet, or what was that? The gray?
Oh, the countries that were in gray, those are countries that don't plant any genetically modified foods. So it's banned, or it hasn't been brought for approval. So there's two different things. It may have been brought for approval and it was turned down, or those are countries that may not have applied for approval to grow those crops. Okay, there is all of the what is a genetically modified organism, what are the types of genetically modified organisms that we're likely to find out in the field, how much of it is planted, who plants it, those sorts of pieces. At this point, I'm going to transition, and we're going to talk about what do people say and think about genetically modified foods. And we're going to spend the bulk of our time in this part of the conversation. I have this picture up here for a reason, because on the surface, if you start asking questions about genetically modified food and you go to your favorite search engine and you start looking at web pages, right off the bat, you're going to be led to believe that the argument about genetically modified foods is very simple. It's a yes or a no. And depending on what website you go to, the yes is because they're the best thing ever, or the no is because they are the most horrible thing and they are killing all of us and our children. In reality, the conversations about genetic modification and genetically modified foods are not black and white, but are much more gray and nuanced. So I almost titled tonight's talk GMO 50 Shades of Gray, but decided that that was probably not the best approach to take. But the point is that whatever you, let me say that differently. It's very difficult to blanketly say they are all fantastic and benefit the world, and it's very difficult to say they are all awful and should never be brought to commercial value. And I hope as we walk through the next hour, some of that becomes clear. Now, I've said from the very beginning, my goal is not to try to drive a specific agenda with you. I am not going to tell you you need to walk out of here saying, I'm going to go buy genetically modified foods. Nor am I going to tell you you need to walk out of here saying, I'm never going to eat genetically modified foods. But my goal is to try to cut through all of the hype and all of the vitriol and all of the shrill and try to give you at least a fair sense of what are both sides saying and what is the data showing us. And then let you make a decision. That's the goal. That's the purpose of what we're going to talk about. There's a great set of recommended readings if you want lots of detail. And that's a series of articles from a magazine called Grist. And this series of articles, there are 30 articles in this series called Panic-Free GMO. Now, Grist is an online, environmentally minded magazine that generally tends to be a little liberal-leaning in many of its writings. This was an incredibly well-balanced series of articles. And judging by the number of comments that each one of the articles received, which generally ranged between 300 and 800, there are people on both sides of the argument that were angry, which means this guy probably hit it just about right. because there were some things that the pro side loved and the anti hated, and then the next issue, there were things that the anti side loved and the pro hated. So just recognize, if you want more reading, this is a great place to begin your conversation. If you'd rather not start here and you'd like to use your favorite search engine, well, have at it, but you are going to spend days and days and days digging through some of the content. I want to start, before we get into any questions, with what I think is really valuable as a starting place. So Nathaniel Johnson is the author of all these articles from Panic-Free GMO. And he says this: notice the ground each side has staked out as their presumptive starting place. So what is the perspective that the two sides are coming from? And I think this is a really valuable conversation for us to have and for you each to ask about yourself. You have one side that sees humans as fragile and dependent on maintaining the nurturing environment in which they evolved. In other words, it is very, very important that we make sure that we do everything we can to protect the world that has brought us to this place and not to allow change. The other side sees humans as tough survivors of a fundamentally chaotic environment. The world is continually changing, and we have had to pull and scramble and fight our way to survival, and it is us against the world. One side sees huge dangers in technologies that alter our surroundings, which makes sense because it is really important to maintain the environment in which we evolved, to keep it as unchanged as possible. The other side sees technological advance as a defense against nature, red in tooth and claw. I love this phrase, red in tooth and claw. It was first coined back in the 1850s, and I think it also shows up in a poem by like T.S. Eliot, but I'm probably wrong about that. So you can fact check me if you want. But anyway, the concept of nature red in tooth and claw is the concept of nature is wild, and it is ready to get us. And it's going to get us, and it's going to tear into us, and it's going to have blood on its teeth and blood on its claws as it just destroys us. So, and there are both of these. Sometimes the earth is good and nurturing and kind, and sometimes the earth just smacks us down. So there are truths in both of these perspectives. But I want you to think about these perspectives throughout the rest of our time together in this conversation, because this is the place where these two sides draw their energy or their strength from. Is the earth something that we have to protect and technology threatens that? Or is this about our ability to overcome the challenges the world throws at us as it continually changes, and these new technologies represent our triumph over that? So those are very, very different perspectives. All right, question number one. We're going to do this in this kind of a format. There's going to be a question. Are GM foods different from other foods? All right, and I'm going to show you in each one of the slides, I'm going to show you the pro-genetically modified version on the left, and I'm going to show you the anti-genetically modified version on the right. And the use of color is totally arbitrary. I'm not trying to signify anything by saying that one group is green or green is good or blue. I'm not going down that road. So, are genetically modified foods different? The pro-GM position is they're no different than 10,000 years of traditional breeding. The anti-GM position is they are dramatically different, and they contain multiple unknown risks. The pro-GM side, oh, it was from Tennyson. 1849. Thanks, Michelle. All right. The pro-genetically modified side says that DNA is the common instruction across all living organisms. So the DNA from a bacteria is read and interpreted no differently than the DNA from a human or from a corn plant. The anti-GM side says, well, okay, DNA may be DNA, but species don't crossbreed, and there's a reason why they don't crossbreed. And just because something works one way in a bacteria is no guarantee that it's going to function in that exact same way in a plant, even if DNA is DNA. And these viral and bacterial switches, they have completely different functions potentially in other organisms. We know what they do in bacteria, but we don't know that that necessarily means they're going to operate the same way in a plant or in another animal. The pro-GM side says, yeah, but these have been with us for thousands of years, and as a species, we've interacted with them, and we've ingested them when we've eaten them on food and when we've eaten them in dirt. So if they were harmful, we would have known that by now. The anti-side says we've only had 30 years to assess genetically modified technology. That's not necessarily enough time to try to give us long-term effects. And the concept of an additional risk, no matter how small, has been placed on us without asking our permission. So nobody asked if I wanted genetically modified corn in my tortilla. But there it is. And even if it's a small risk, it's a risk. Okay, just with this storyline, do you see those two different perspectives that we talked about? That whole concept. There are risks, and those risks, they may be small, but they are still risks. Oh, but this is technology that we've worked with, and this technology is using DNA, which is the same recipe. All right. Periodically, I'm going to give you my commentary on these positions. Not always, though, so that'll probably frustrate some of you. But if we ask the question, is it different? In reality, it probably is different. This is different than traditional breeding because we're pulling in information from another species that we wouldn't naturally be able to assess. Is it that different? No, not really. In fact, it's probably less of a concern than when we irradiate seeds and we get lots of mutations and we don't know where those mutations are. Because this technology is at least precise and I know the pieces I'm working with. But is it the same? I think it's a little bit different. Do we worry about how little that difference is? I think there's the question that we have to try to get our arms around. How are genetically modified foods regulated? All right, look at this. Anti-GM, they're not required to do any safety testing on genetically modified foods. Pro-GM, genetically modified foods undergo an enormous amount of regulatory testing. Okay, this is an example, and you're going to see this a lot tonight, where one side says the answer is 5, and the other side says, no, the answer is blue. Sometimes the answer is 5. Sometimes the answer is blue. Sometimes the answer is neither, and sometimes the answer is both. And this is an example where the answer is actually both. These groups are both right, but it depends on the definitions of the words they're using. And that's where it gets tricky. So, for example, testing of genetically modified foods involves at least 2 and sometimes 3 regulatory bodies in the United States, and there are different regulatory bodies in other countries as well. The U.S. Department of Agriculture has to be involved, especially when these are being field tested and before it can lift the regulations around field testing. And because this is a food, the Food and Drug Administration has to be involved. If this is a genetic modification that produces the Bt insecticide, the EPA is also involved, because now we're dealing with a pesticide and what is its impact on the environment. So there are 3 different regulatory bodies that have to look at and check off on genetically modified foods in the United States. But the process is voluntary. Okay, what does that mean? How do you respond back, pro-GM side, if the process is voluntary? Well, the pro-GM side would say, well, yeah, the process is voluntary, but everybody volunteers, because nobody is willing to go up against the FDA, because if the FDA says, nope, we don't like this, they're going to ban your food. So even though it is not required, everybody volunteers. So right there, depending on the definition we use, it is not required, but there's an enormous amount of it. So do you see how each side is right, depending on the perspective that they're taking with this? The requirements for safety testing are determined on a case-by-case basis. So how I'm going to determine if genetically modified corn that has Bt, the Bt pesticide in it, is safe is going to be very different than the way that I'm going to determine if a corn plant that is drought tolerant because it has a gene from bacteria in it. So it's not the same regulations plant to plant. And that makes individuals nervous because there's no consistency. The pro-GM side says that actually is a benefit because we can specifically hone in on the specific compounds that we think are most relevant based on what we know about this genetically modified plant. If we have a standard set of molecules that we have to test, and we have a genetic modification that doesn't change any of those molecules, then I have no way to know its validity. So there's the other side of the argument. Sometimes it feels like groups are talking past each other, or that there's a set of specific, when I hear you say this, I say that. All right, so I think it's fair to say they are regulated. There's a fair amount of regulation. There are some concerns about there not being a standard set of regulations, but I think there are some valid points why you want to have flexibility in this process. Are there health impacts to eating genetically modified foods? We're going to look at this at several different levels in terms of what do we mean when we say health impacts. The pro-GM side says genetically modified foods have no higher risk than conventional foods. The anti-GM side says we don't know the health risks associated with genetically modified foods. We just don't have enough information. They might be small, they might be big, but we don't know. One of the things that has been raised as a point of concern is, can the genetic information, the resistant gene, the herbicide resistant gene, the drought tolerant gene, can that actually, as I eat that food, can that somehow get all the way down to my digestive system and get into the bacteria in my gut? And is that something I should be concerned about, especially if one of those switches is resistance for an antibiotic? This concept is called horizontal gene transfer. And horizontal gene transfer is mentioned not only in terms of can this get into my gut, but out in the field, can this gene get out of the plant and get into another plant? So horizontal gene transfer.
And do you know if any of that has actually happened?
The data that has been looked at so far, at least in the gut, suggests that that has not actually happened. The trick with that, David, is saying it hasn't happened doesn't mean that it means that I have not been able to detect it happening. So we're back to this issue of risk. If I don't see it and the risk is small, are we willing to live with a small risk? And how do I quantify what a small risk is? Now, we certainly know that there are examples where bacteria transfer genes all the time. So we've seen horizontal gene transfer naturally happen. So it is something that we know happens in the world around us. The pro-GM, one of the arguments is so processed food's already been processed in such a way that much of the DNA has been broken into smaller pieces, and when you eat food, it's been processed even further. Well, the EPSPS gene, which is the gene I told you that confers resistance to the herbicide, has actually been shown to survive through the digestive system intact. So it didn't get chopped all the way up. Well, it's highly unlikely that that naked piece of DNA is going to actually get into a bacterial cell and be incorporated into the nucleus and then be active, because bacteria have a natural defense mechanism that chews up any foreign DNA that comes into its cell. So the likelihood that even if it could get in, that it would make it intact is very, very small. And the EPSPS study used individuals that had serious medical issues and potentially something not quite right about their digestive system. So when they looked at individuals that had complete intact functioning digestive systems, they didn't see the full transgene make it through. Well, most recent work has found that DNA from food actually circulates in our bloodstream. So if you have a hamburger and I take a blood sample several hours later, I'm going to find beef DNA. Okay, well, that's pieces of beef DNA that somehow does get into the bloodstream, which wasn't something that we necessarily thought happened. But back to the same storyline, is that going to necessarily get into your cells? And what is the likelihood of that? Probably very, very small. I can't say it's zero, but it's probably very, very small. How comfortable are we with very, very small? What's our tolerance for risk? Here's a second one, allergens. You hear this one a lot when we talk about genetically modified foods. We don't know the health risks associated with genetically modified foods because it could be that the gene that I'm bringing in from another plant or from another animal actually is going to create a protein that's going to cause an allergic reaction in an individual. So who's to say that genetically modified plants won't cause allergy? Well, the current testing regimen screens for known allergens pretty thoroughly. And in fact, there was an example, I think it was corn, that used a gene from the Brazil nut and actually was found in the regulatory testing to be capable of generating an allergic reaction. So it was never brought to market. So the system works. It recognizes allergens. Yeah, but the system works because it screens for things that we know cause allergens, like egg and soy and peanuts. What about the stuff that we don't know couldn't actually be an allergen? The system doesn't screen for that. It's the unknown unknown. We don't know what it is. And so the very example that we picked it up in that Brazil nut doesn't necessarily mean that there wouldn't have been another protein that we wouldn't have identified. Well, okay, but the gene that we have brought in makes this protein at a very, very low level, generally below the level that scientists and physicians say is necessary to be prevalent enough to generate an allergic reaction. So even if it were an allergen, it's in such minute amounts, it shouldn't be a problem. But we don't have the right test to tell us about everything that could be an allergy. This, I think, is a really valid point. This is a concern. We don't have a good way to test for the unknown allergens. The problem is there isn't a really good way to test. So the American Medical Association in their 2012 position paper says it should be noted that absolute avoidance of all risk is not achievable. We can never make this zero risk. And that's true not just in this, but in just about everything we talk about. We can never eliminate all risks. We can minimize them to an acceptable level. And what is an acceptable level? Research, this last sentence, research to examine more effective methods of allergenicity assessment is ongoing. So there are lots of technologies under testing right now to try to identify potential allergens, even if we've not seen them before. There are some differences of opinion about the technology that we use. I think everybody seems to agree that they're not ideal technologies. So if we codify them, if we regulate that this is the technology we should use, what does that mean when another technology comes along and it becomes really difficult to rewrite the regulations? So we're in this kind of limbo phase. I think that this is an important issue that we need to pay attention to. All right, how about long-term effects? What's the impact of eating genetically modified foods over the course of my lifespan? The pro-GM side will say, there was a really rigorous health analysis that was done in order to bring any genetically modified food to market. So laboratory animals are fed relatively high doses of this food. They're monitored. There's a control group of rats or mice or whatever that is fed the exact same diet. So if it's genetically modified corn, it's the same strain of corn minus the genetic modification. So they've tried to control for everything they possibly can, and there have been no health effects that have been seen in that window in any of these studies. Well, the current safety tests only run for 90 days. So there may not be any acute issues, but are there long-term issues? And a 2-year study, long-term, feeding genetically modified corn to rats discovered that the rats developed tumors, and that the tumors that those rats developed were so big that in some cases it killed the rats, and in other cases the rats had to be sacrificed because of the tumors. Okay, I did not show you pictures of these tumors for a reason. They are really disturbing, but if you want to go Google them, you can find them with whatever your favorite search engine is. That was a powerful study when it came out, and it was a firestorm study. The anti-GMO end of the room said, aha, here you go, long-term effects. And the pro-GMO side said, wait, how did you do this study? What did you do? You used Sprague-Dawley rats, a specific breed of rats. Sprague-Dawley rats develop tumors as they age. And so the controls also develop tumors. And they developed a lot of tumors. Maybe not quite as many, but enough to say, can we determine that the tumors are because of the GM food or just because of the rats that we used? So we don't think this paper is scientifically valid, and we think that this paper should be retracted. And there was such an uproar about it that the journal retracted the paper on Thanksgiving Day, which is a really interesting... But they retracted the paper, and they said, we're retracting the paper not because the data was falsified, not because there was any forgery or mishandling of the data, but because we can't make enough decisions based on that. So this is a firestorm, this back and forth debate over these tumors and over these rats. And again, depending on the point of view you start your conversation with, you're going to find a piece of information that will support either side of this argument. There was a paper that came out at the end of last year that said rats fed genetically modified corn developed tumors, and they used a different variety of rat. But the control rats were fed wheat. That doesn't help. It doesn't do any good. And then you also have to follow the funding, because a lot of these studies are funded by the groups that want you to arrive at one conclusion or the other. They're not independently funded, or they're not peer-reviewed. They're put in a journal where you pay a certain amount of money and it shows up in the journal. Other scientists didn't look at it and dig through the data and kick the tires. So this is a real issue. The pro-GMO says we have fed trillions of meals with genetically modified foods, and we see no effects. And the anti-group says, well, if this was only slightly increasing the incidence of a very common and long-term disorder, it'd be really hard to track that back. If you go search on the website, you're going to see all kinds of great correlations. Remember what we said in week 1? And we said this in the microbiome session the year before, every single week. Correlation does not equal causation. It doesn't rule it out. It could be the issue, but just because it's correlated doesn't mean it is what causes it. So there are all kinds of gorgeous graphs that show you the incidence of genetically modified crops and the subsequent incidence of the spraying of the herbicide glyphosate. And then right against that, they match the incidence. The most current one is the incidence of autism, and they match really nicely. Or the incidence of kidney cancer, or the incidence of Alzheimer's. Those are correlations. But I could also map the correlation of the increased use of cell phones, or of portable electronics, or the increased consumption of organic foods. And I don't think the increased consumption of organic foods is causing the increase in autism. I think correlations are something we need to look at, but in too much of the current literature, correlation is being taken by the public as causation. Oh, look, they've increased the amount of herbicides they've sprayed on food, and we got all these new increases in diseases. Never mind that from the 90s, the early 90s to the late 90s, we completely changed the definition of what autism was, and so we opened up the diagnosis to many, many more individuals. But you got to dig beyond this, and most of us don't have the time or the background or the stamina to do that. So be careful with some of these pieces. Again, I'm not saying long term, over 50 years. We haven't been eating genetically modified food for 50 years. So we need to pay attention to that, but all the studies that have been done, the long-term ones in model animals, that are done with the right kind of controls haven't turned that up. And believe me, there are a whole bunch of scientists that would love nothing more than to be the one that finds the cause of autism. So those issues are there. Are there environmental impacts? Super weeds. Super weeds are weeds that have gained resistance to herbicides. So here's how this works. So DNA mutates all the time. We've talked about that week after week. And sometimes that mutation happens in genes that lead to a change in physical trait. So by chance, I may have a mutation in the gene in the EPSPS gene of corn that randomly makes it now resistant to the effects of herbicide. So I can spray the herbicide, and because of this mutation has now made the herbicide ineffective. So there's the mutation. It occurred randomly. If there's a selective advantage for it, so all the other weeds die, but it survives, it spreads its seed, next year I'm going to have a whole lot more of those. And over time, I'm going to see an increasing population of that specific genetic change. That's the whole process of selection. A mutation randomly occurs, and if the environment is favorable for that, then I'm going to see that at a higher frequency. It's going to be passed on to the next generation. So super weeds have happened since we started spraying herbicides. We've got lots of super weeds that don't necessarily have anything to do with genetically modified plants. So that's an argument that this side of the table makes. This side of the table responds back and says, yeah, that's true. But because we have so many genetically modified crops that now are resistant to the effects of glyphosate, the active ingredient in Roundup, farmers plant Roundup-ready crops year after year after year and spray glyphosate on their fields over and over and over. So they've created conditions that are ripe for the emergence of weeds that are resistant to glyphosate. Because if a random mutation occurs, we are certainly in an environment that is conducive to those resistant genes taking root and growing. So genetically modified crops may not be the cause of superweeds, but our reliance on only spraying for a specific type has given rise to herbicide-resistant, glyphosate and glufosinate-resistant weeds. Well, diversified weed management is the key to controlling that. And that's a fair statement. Both sides of the table agree on this. You want to rotate out the kind of things that you spray. You want to, in some cases, till the soil under. You want to plant ground cover crops periodically. You can't rely on only one type of weed management, or you increase the likelihood. And scientists have developed additional genetically modified traits that now give us resistance to this herbicide and that herbicide. So we can stack the traits, and a farmer can spray 2 or 3 different kinds of herbicide, and the chance that any one weed is going to have developed all of those resistances is really small. So remember, back to our storyline, technology helps people triumph over a continually changing world. We'll make more genetic modifications. There's part of where that storyline comes from. Well, ultimately, that means that farmers are using additional herbicides, which may not be nearly as less toxic as glyphosate. Glyphosate is one of the least toxic herbicides there is. But if we now develop lots of weeds that are resistant to it, and we have to go back to some of our older herbicides, they were a whole lot more harmful and a whole lot more damaging. There is a continual balance point between whether we're talking about bacteria or viruses or pests. Pests figure out how to take over a cell, how to infect a cell, how to eat a plant. And then the plant has mutations that occur that give it an advantage again, so it gains the upper hand. So it produces something that's actually toxic, or it figures out a way to evade infection by the virus or by the bacteria. And then as a new mutations arise back in the other population that now lets it get around. So it is this continual arms race between whatever you're talking about. And so this is an arms race between resistance and technology. And new resistance comes up, and we have to develop new technology. And the argument on this side of the table is how much are we going to escalate that arms race? And the argument on this side of the table is the technology is always going to let us develop ways to stay one step ahead. Here's another side where both sides are saying 5 and blue, and they're both right because of their definitions. The anti-GM side says genetically modified crops have increased pesticide use. And the pro-GM side says genetically modified crops have decreased pesticide use, and they've also increased the capture of carbon. So we're reducing our carbon footprint. These groups cannot both be right, but they are, because it depends on the definition that you're going to use. This side talks about, because of Bt corn and Bt cotton, we no longer have to spray insecticide on those crops. So we've taken a pretty significant amount of insecticide out of the soil, out of the environment. And because I'm not spraying as much, I have fewer planes that are spraying, fewer trucks and tractors that are spraying. I'm putting less fuel into those tractors. I don't have to turn the soil over, which was a common way to get rid of weeds. So I'm not releasing carbon into the environment. So this has actually been pretty impressive. I've removed the equivalent of 12 million cars off the roads in terms of my carbon and emissions capture. This side of the house looks at the data and says, well, you may have reduced the insecticide that you've sprayed, but you have increased the herbicide so much that you've totally wiped out the gains that you've just described. So if you look at these are the percentage of U.S. acreage planted with genetically modified soybean, cotton, and corn. And here's when the Roundup Ready crops, the genetically modified crops, were introduced. And the percentage of crops has gone up dramatically, and the overall amount of herbicide that's been applied to those crops has also dramatically increased. So we're spraying less insecticide, but we're spraying a whole lot more herbicide. Now, we're spraying the herbicide because this makes the farmers, this is beneficial to the farmer. I'm not trying to give the farmer a bad rap here. This makes it much easier for them to control at least an element of their risk by having plants that, in a year where corn or cotton borer worms are bad, this actually gives them some protection against that and helps maintain their crops. Or it helps them control the weeds and the timing of how they spray. But do you see how both of these groups are right based on the definition that they're using? And they're looking at it through very different lenses. Okay, let's do this. You've been sitting here for an hour and 10 minutes. I want you to close your eyes for a second. Those of you at home, play along with us. Close your eyes and take a deep breath. Take it in, 1, 2, 3, 4, and release it. 1, 2, 3, 4. Do another one in, 1, 2, 3, 4, and another one out. 1, 2, 3, 4. Now keep on breathing at whatever pace you want, and I just want you to just tense up your shoulders, your neck, your arms, and then relax them. And tense them and relax them. All right, open your eyes. Okay, a little mental break is always nice when you sit in a room listening to one person blabber on and on and on. So now I'd like you to turn to your neighbor, and I'd like you to say to your neighbor, "Neighbor, if you weren't here, I'd be the best looking person in the room. If you weren't here, I'd be the best looking person in the room." All right. Little mental break. Let's dig back into genetically modified organisms. Impact on biodiversity. Genetically modified corn pollen is toxic. Bt corn pollen is toxic to monarch butterflies. And the decline in the monarch butterfly population can be attributed to the production of Bt corn and the pollen. In laboratory tests, if you spread Bt corn pollen on milkweed leaves, the caterpillar, when they eat the milkweed leaves, ingests the Bt cotton, and it's toxic. The toxicity levels in the test were about 5 times higher than what you would expect to find in nature. And only during a brief window of time when the cotton is producing pollen and it happens to overlap in certain geographical regions where the monarch butterflies are spending the summer. So yes, Bt pollen is toxic, but not toxic at the levels that you would experience out in the field. Okay, well, increased herbicide spraying, because we now have herbicide-tolerant plants, is killing the plant that the monarch butterflies lay their eggs on and that the caterpillar need for food. Yes, that is exactly true. But that plant is milkweed, and it is a weed. And it's not the genetically modified crop fault that it is killed by the herbicide, it's a weed. If we want to solve this problem, we need to plant refuges, large areas of milkweed that are not going to be sprayed, or we need to have people plant milkweed at home. And if you're going to plant milkweed at home, you need to do a little bit of digging, because there are certain varieties of milkweed that are most commonly available by mail that are actually not good for the monarchs. So you need to buy the right kind. So be careful, because there's been a study that was released that says that in our good intentions, we actually may have made the problem worse the last few years because we've planted the wrong kind of milkweed. But again, here's this argument. Yes, we are getting rid of the milkweed, the monarch's food supply, but that's because it is a weed and we are increasing spraying of weeds. So yes and yes, what's the solution? Rather than just yelling. Bees. How about bees? Yeah, bees. B-E-E-S. So hive collapse. And is the hive collapse caused by genetically modified pollen? There are a whole, all right, so now I'm going to say that was a topic that I looked at, and I don't have time to cover that, so I'm only going to glancingly talk about it, so we're at the edge of my knowledge. There are a number of different things that are currently being investigated as what is the cause for hive collapse. It's a critical issue because we are losing more and more and more of the bees that pollinate most of these crops. And we're now actually having to have farmers hire beekeepers to bring hives into the middle of their fields to pollinate during that critical time. So there are a number of different things that are being looked at. I know that there was a lot of suspicion pointed at genetically modified crops. I haven't seen anything. What I've seen looks a little bit like the monarch butterfly. At really, really high levels, it's toxic, but is that what we're likely to find in the real world? So I can't give you a definite answer one way or the other. But yes, bees is another piece of this whole puzzle.
Are farmers locked into using the seeds, and are the seeds like licensed to the farmer to?
Yeah.
So in other
Are the
words,
farmers locked in to the seeds, and do they have to sign an agreement when they buy the seeds?
Right.
There is an agreement that they sign when they buy the seeds. There's a whole set of things that they agree to in that agreement. One of which is that they're not going to harvest and keep the seeds to replant for the following year. Now there's some variation on that depending on if you're talking about humanitarian needs in like Africa. So we're going to set that to the side for a minute. Depending on the type of crop, it is almost always more expensive than non-genetically modified. And in some cases it can be as much as $100 a bag more expensive. There is an agreement that they sign that is a binding agreement. And there have been issues in the past with individuals that bought genetically modified crops one year, didn't buy them the next, and when they came out and looked at that individual's crop, they had saved their seed and they were replanting. And then there's a lawsuit that's involved. Because that's part of the agreement, that there is intellectual property that the seed company, and Gary, we're going to come to the seed companies in just a second, so hang with me. But the seed company has intellectual property in building this. They've invested a lot of money, and they're not just going to give it away to you to use year in and year out. It's a little bit like, from their perspective, it's a little bit like music piracy. Like just handing somebody, you bought the download from iTunes and then you distributed it to 15 people. There are licensing agreements. So let's come to that. GM crops lift small farmers out of poverty. GM crops are about patented ownership of the food supply. Very different perspectives. GM seeds are far easier to patent. I just told you that. And all GM plants have patents on them. Let me rephrase that. I'm sorry. Most GM plants have patents on them. There are some GM plants that are actually produced by nonprofits, and they may still have a patent, but they may be giving the seed away at regular cost, or they may have found other ways to fund it. So everyone who is working on genetically modified crops is not necessarily part of the large agricultural companies. So recognize that. 10 companies control 2/3 of global seed sales. I think that is a statistic that we need to pay attention to. The majority of our seeds are in the hands of a small number of companies. And how we deal with that and what that means for seed security also depends on the perspective that you bring to the table about your thoughts on what's commonly called big agriculture or big A. And is that okay? Does it need to be distributed into multiple hands so that we resist? Do you worry about monopolies, about a small number of groups being able to control who gets the food, who has access to the food? Everything that I'm putting on here on all of these slides are pieces taken directly from position papers, from briefs, from background, from both sides. So this is not anything that I'm using my own words. I'm using their background, which is why you've got these references. GM Watch says that genetically modified seeds chase away the competition and coerce governments into surrendering control. That is a perspective. That is a viewpoint that is held somewhere on that spectrum. Pro-GM says Biotech cotton, BT cotton, significantly lifted over 16 million resource-poor farmers out of poverty. Okay, how can that be true if BT cotton costs so much more? So we've got two seemingly divergent arguments. And now let me take you down the rabbit hole of the nuances. I've not done this on all of our other scenarios, but every scenario has a storyline like we're about to walk through. So hang with me.
Don't these patents expire after some number of years?
The patents do expire after some number of years, but because they're continually creating new versions of the crops only stay on the market for 3 or 4 years, and then they're replaced by something that has multiple resistances or stacks 3 or 4 things. And that's part of the challenge in trying to do the testing, because things aren't on the market necessarily long enough to do good comparisons. We keep the variables keep changing, or progress keeps marching on, depending on the perspective that you're taking from it. Okay, so here's the financial impact. This is looking at a meta-analysis, so looking at about 8 or 9 different studies looking at the financial impact of genetically modified crops. This is a big business. $15.7 billion US is the global value every year of biotech crops. And of the money generated by genetically modified seeds, this analysis, looking at multiple studies, found that 10 to 70%, first of all, 10 to 70%, 10 to 70% of the benefit is captured by the farmers. So the farmer actually reaps the benefit in larger yields or in less costs for spraying. Now, some of the farmers probably are laughing at that 10 to 70%. Many of them, it's much closer to 10%. And then, so there are the percentages of the benefit, in other words, the savings or the increased profits. 10 to 70% from the innovators, the people that make these. And then what's left, 5 to 50% of the consumer in lower food prices. These numbers are they're so broad that they don't really it's really difficult to ascribe any kind of meaning to that. Because if the farmer's getting 70%, that's one thing. If the seed company's getting 70%, that's another. If the consumer's getting 50% of the value passed on in lower food, or if the consumer's getting 5%, those are very different storylines, depending on how we interpret who makes this. I think the point we can take away from this is that it varies dramatically. And if you look at this, many of these studies were 96, 98, 99, 2001. They haven't been recent studies. I think it's time to do those kinds of analysis again. Are genetically modified crops required to feed the world? All right, here's our rabbit hole. GM crops increase yields. GM crops do not increase yields. Both are right. GM crops do not have a higher intrinsic yield. So there are not more kernels of corn on a genetically modified corn plant. There are not more stalks of corn. There are not larger squash or larger papayas. The intrinsic yield, the amount you get from a plant, is not dramatically different. But if you look at the overall yield from the harvest, it is larger, especially in the instances of BT, of insect-resistant corn and cotton. So because you are having less of your crop lost to worms, you're capturing more from the acre that you're farming. Now, that may be, so from this study, BT cotton, 33% increase in yield, no increase in yield, 24, 37, 9, 22, 10. The numbers are pretty broad, but there is an increase in yield, at least with BT. With herbicide resistant, there may or may not be an increase in yield. Some of it depends on the amount of weeds that you would have had to have dealt with that you don't have to deal with because you can spray glyphosate on it. And it makes the farmer's life easier because there are less things they don't have to go out and individually spray the weeds or till the weeds over. They can pass over it once. It is a much easier decision, and that easier decision gives them time and energy to have to deal with something else with farming. So there's a benefit to that. It may not be an increased yield, but there is a tangible benefit to a farmer with an herbicide resistant. The anti-GM argument would be, well, yeah, the yields of all crops have increased, but they've increased because we have better farming practices. We know more about cultivating the soil. We know more about irrigating the soil. So that really doesn't have anything to do with genetically modified crops. And then the pro-GM group says, oh yeah? Well, let me show you this. This is an image of BT, of cotton crops in India. And along the x-axis is number of years, starting in like 1980 and running to 2010. And this solid line is the general increase in yields until we hit this dotted line, which is the increase in yields once we had BT cotton in place. So look at that dramatic increase in yields once we brought BT cotton in. And the anti-GMO says, oh yeah, well I can show you that same graph. So this piece right here, here is that piece right here, okay? And look at this. This is the percentage of the cotton in India that was actually BT cotton. So when we had this huge increase in yields right here, we had almost no BT cotton. So that has nothing at all to do with BT cotton. And then the pro-GMO side said, oh, but not so fast. Because during these years when BT cotton was not official, there was a whole lot of illegal BT cotton that saturated the market. So yeah, it actually is BT cotton. We just didn't see it. Then, and by the way, the increased debt from having to spend more to buy the cotton seeds to begin with, and then having to take care of the cotton, means that our farmers in India are having to go into debt to borrow money, and the stress of that and never knowing if they're going to have increased yields or not is increasing the number of suicides in India by tens of thousands every year. So genetically modified crops are killing people around the world. Okay. Well, here's a graph. Again, this is a correlation. Here's the increase in BT crops. There it is. Now, this increase could be, we could be masking the illegal BT crops, so we'll own that. And here is the rate of suicide among farmers in India. So it's actually gone down a little bit. Okay, this, and again, I now realize I've crossed over into the facetious, and I apologize because farmer suicide is not something that to laugh at. It is a serious matter. Regardless of what the trend in these numbers are, there are a number of farmers that continually feel that they're never going to make ends meet. So I don't mean to, I apologize, I don't mean to belittle or laugh at that. But this is the level of argument that you find in trying to understand, trying to untangle these storylines. Oh my goodness, look at what time it is. All right, food security, we're nearing the end. Pro-GM says that genetically modified foods are a key part of feeding the world. In fact, some groups go so far as to say we will never feed 9 million people unless we rely heavily on genetically modified foods. On the other side of the spectrum is there are much better ways to feed the world, and we have spent so much money developing genetically modified foods that have only given us marginal yield increases and mostly have dealt with herbicide tolerance that we could have put that money to much better use for other traditional breeding and other farm cultivation practices. So they suck up all the money in the research world. And that's pretty much what I just said on both of those sides. So where do they fall? People find themselves on all parts of this solution. Clearly, we are going to have to find more food for a growing population. The question is, how much of that is GM? And when you get down to it, even most pro-GM people will say, genetically modified foods are one solution, but they're only one, and there are multiple solutions that we have to embrace. We cannot simply say it's all going to be genetically modified food. Should genetically modified foods be labeled? Yes. Let's take a poll. How many of you think genetically modified? No, I'm not going to do that. Never mind. We're not going to do that. Okay, here's the pro-genetically modified argument. The pro argument is the FDA have said there's no difference between genetically modified food and non. So labeling them would imply that there is something different and imply that there's something hazardous when we have no evidence that there is anything hazardous. In fact, we have no health studies that have been well done and rigorous and peer-reviewed that say that there is anything wrong. And if we're going to label them, we're going to have a whole bunch of individuals that can regulate all the different foods and say, was there any genetically modified food that went into that Dorito? Was there any genetically modified corn? Do you know all the suppliers? What are they planning? Can we verify what they're planning? And that's going to increase prices. And the people that are going to bear the brunt of those prices are the working poor, because they don't have the income to spend more money to buy organic food. There's the argument. It's a passionate argument. There's an equally passionate argument on this side, and it's very simple. People have a right to know what is in the food that they eat. All right, here's my commentary. I actually think that foods should be labeled. I think foods should be labeled not because I think that they're dangerous, but I think people need to know, and I think people are afraid because they don't know if it's in their food or not. And if it's labeled and people make choices, and over time we see that there's no difference, then the label loses its scariness. It loses its fear. I recognize these issues, and I think there are real concerns with how much it costs to regulate this and where does that put the burden. I think that is a concern. I think that's a worry. And I recognize right now that there are foods that you can buy that say GMO-free verified. So you've got options in buying foods that are not verified. In the same way, the GMO-free, panic-free GMO series makes this analogy. If you go into a restaurant, do you have a right to know if your food has been prepared with pepper in it because you're allergic to pepper? Yeah, I think you have a right to know that. Do you have a right to know if your Brussels sprouts have been harvested by individuals in Central America that are paid a living wage? I don't know where that line is. What is your right to know? How much right do you have? But I personally think there is some real value in seriously giving thought to how do we look at labeling so people know. Because right now, there's a lot of unknown, and unknown makes things scary. Are GMO crops safe? There are lots and lots of groups that have looked at this. 6 ways to Sunday. Let me just cut to the chase. Here are 10 different scientific organizations that have said genetically modified crops are safe. These are people that I trust. These are people that have looked at the evidence, like the World Health Organization, the European Commission, the American Medical Association. I recognize that on your handout and on this screen, it's really difficult to read this, but if you go to the Genetic Literacy Project and you look for GM infographic, you'll see this and you can read it. You can blow it up and you can look through it. But here's the challenge. So we have a whole group around the world of scientific agencies that say genetically modified foods are safe and they pose no health risk and they're no different. And we have a population that says, I don't believe you. I think it's quite different, or a portion of the population. Why is there such a disconnect between our scientists and the rest of the world? Now, I think, honestly, part of it is the scientists' fault, because sometimes, not all scientists, believe me, I'm doing major generalizations, but historically in the past, we've been a little patronizing about this. They're there, it's all right, you need to trust us, we've looked at this, it's safe. That's not a way to engage in dialogue. That shuts down dialogue just as much as, you're killing my children, shuts down dialogue. But here's why I think part of why we struggle with this. This is a great article written by this guy, David Ropick, in Psychology Today about 5 years ago. And he talks about risk perceptions for genetically modified foods. And I've broken it down. I've kind of chunked it into smaller pieces just to make it easier to handle. But he says the perception of genetically modified food is like the perception of any risk, a combination of the facts and how those facts feel, a mix of reason and gut reaction. So if you think about that, any decision that we make, we know the facts, and we also know how those facts make us feel. I'm going to get in my car. Many of you are going to get in your vehicle that are in this room after we're done. And you know that there are risks with getting in your car. But you also know how you feel about those risks, and you've decided it's okay to get in the car. Some of you go skiing. There's a whole lot of risk associated with that. But you know how you feel about that, and so you've blended the fact and how you feel, and you've decided to go skiing. Some of you go bungee jumping. Some of us don't. The facts and how you feel about the facts drive that. Genetically modified food has several characteristics that psychologists have determined make some things feel scarier than others. So psychologists have identified what are those things that make us anxious. Notice they said this word right here, feel scarier. They didn't say it is scarier. They said they feel scarier. I'm in the close wrapping up, so for those of you that are watching the time, hang with me 5 more minutes. It's human-made, and that alone makes it scarier than a risk that's natural. We made it. It didn't grow naturally. Part of the argument I'm trying to help you see, not part of the argument, part of what I've tried to help you see is that natural has been very man-driven, human-driven, for 10,000 years. We're more afraid of what we can't detect ourselves. I don't know if there's genetically modified food in this. I don't know what it contains. I don't know if it's got an antibiotic-resistant gene in it. I don't know. I can't tell. I don't have a handy-dandy sensor that lets me poke the food and go, whoop, GM, GM. We're more afraid of what we don't understand. We've spent 6 hours talking about agricultural genomics. And I think none of us in this room feel like we have a handle on it. I surely don't. And we're more afraid of what we're exposed to involuntarily. It's in my food. Is it in my food? Do I have a choice if it's in my food or not? We depend on the government to keep us safe, but we don't completely trust the government. And that lack of trust feeds greater worry. So, genetically modified foods, even with the facts, have these things that make us more apprehensive. Just by the way we interpret them, we have a greater angst about them, even in the face of the facts. And when you go online and you find all kinds of other bits of information that reinforce your anxiety, that makes it even harder to follow the facts. Very difficult to follow the facts. And these two viewpoints. If you feel that genetically modified foods are part of the wave of technology that helps us feed the world, you are going to come to the table with a different viewpoint, even with the facts and the fear, than if you believe that the technology contains risks. Even no matter how small, risks are risks, and we need to do everything we can to minimize those risks.
You know, there's one item I think you left out, and that is the influence of the media.
The influence of the media. Well, the influence of people that we trust. Whether it's the media or whether it's people that we listen to, we have confidence. And when they say something and we tend to feel that they're telling us the truth, that weighs in how we feel about it. That gives us assurance or it doesn't give us assurance. The media certainly is a part of that as well. All right, let me just tell you this. There are several things on the horizon. Last November and 2 weeks ago, we approved 2 new genetically modified foods. One was called the innate potato, one's called the Arctic apple. They don't brown. Now, they still ultimately rot and decompose. You're not going to have an apple that sits around for 6 months. You will know when this apple is bad, but when you cut it, it doesn't brown. Right now, when you cut apples, they brown through a process of oxidation. If you buy apples in the grocery store that are pre-sliced, they're not brown because chemicals have been sprayed on them to keep them from turning brown. When you go to McDonald's and you get sliced apples, there are chemicals, anti-browning chemicals sprayed on them. These genetically modified foods don't use genes from bacteria or other plants. They use the apple and the potato's own genes to silence the browning compounds. They're still genetically modified, but they aren't foreign DNA. Some people don't like genetically modified foods because they don't like the concept of foreign DNA. If you're still concerned about the other issues we talked about, the issues of agricultural companies and seed companies having control, or where does the money go, that's not going to change that argument for you. But if your argument hinges on, I don't like the fact that it's other organisms' genes, it will change. Maybe. They're creating foods with increased nutritional content. Now, we could talk for a couple days about the golden rice, and golden rice has had all kinds of problems, and the people that put it together have hit roadblock after roadblock and delay after delay. So I am not trying to tell you a rose-colored feel-good story to make you change your mind about genetically modified foods. But golden rice has been genetically modified to contain higher amounts of beta-carotene. So when you eat it, you're ultimately getting more vitamin A in your diet. And people in many countries that eat only rice have a deficiency of vitamin A, and there are tens of thousands of individuals that go blind every year because they don't get enough vitamin A in their diet. So golden rice potentially opens the door to people that only eat rice to prevent that blindness. Now, the first time they created it, it didn't make enough vitamin A to make a difference. The second time they created it, it made enough vitamin A, but the yields, it didn't make enough rice, so farmers weren't going to plant it. So now we're in the third iteration, and we keep saying, golden rice, golden rice. But we have these problems, so let me acknowledge we have these problems, but this potentially changes the life of an individual. So does that change the dynamic for you? Is that different than I have a corn plant that now has this trait? There are brand new editing tools that actually allow you to go in and at the plant's own gene, make a change, and make a change in such a way that it looks no different than if it was a natural mutation that happened. So it looks no different than the mutation happened out in the field, or I irradiated it at this specific site and I got a mutation. And it doesn't leave any switches, any virus and bacteria switches behind. It's called gene editing, not gene engineering. We'll see if these technologies prove useful enough that we employ them, but that potentially doesn't look any different than what would occur naturally, and under current regulations doesn't even fit the definition of genetically modified. So what does that then mean? There are challenges with this, so we've got to be careful with that. But the content's called gene editing. CRISPR is one of those technologies. All right. We have covered way more ground than we normally do on a Biotech 201 night, and you have been incredibly patient and willing to sit through a lot of information. I realize I have minimized conversation and questions at the expense of giving you the breadth of information that covers many of the broad questions out there. So I ask for your forgiveness for that. Agricultural genetics and genomics has incredible power and has incredible opportunities. And there are scientists at HudsonAlpha that are trying to understand the changes in DNA by sequencing these important crops. HudsonAlpha is not creating genetically modified foods, but we are providing the instruction book that lets people understand what these genes are and what they do and changes across different varieties. You will see this field mature and grow dramatically. So pay attention to it, but recognize that the story you hear from the headline is likely only at the surface, and there's much more beneath it, whatever decisions you make. My goal for you was that in these 4 weeks, you would come away much more informed about your food, where it comes from, and the role of genetics in getting it to that point and what is coming down the pike. Thank you for spending the last 4 weeks with me, whether you watched from home or whether you sat in the seats. It is always a treat to be with you. I will let you know in a few months what next year's 201 topic is, and registration will open probably at the beginning of January, just like it has in the past. For those of you that are in the room, safe travels, drive home safely. I think we've got some weather coming at us tomorrow. And thank you for your participation. It's always a treat to work with you.